Hi, today we are going to solve this paper together. I will be doing it real time with you. So we are going to look at the Cambridge International AS and A Level Biology Paper 1, the October-November 2021 1-1 version. So let's look at question 1. So a student observes a cell using a light microscope. The student then draws the cell. Which item will the student need to calculate the magnification of the drawing? So for magnification of drawing, always remember you will need the eyepiece reticule. You will also need the ruler, not hand lens. Hand lens is not a light microscope. It is typically used for plant or environmental field. And also definitely you will need the stage micrometer scale. So this is 1, 2, and 4 for this question. Second question, which equations correctly show the relationship between magnification, image size, and actual size in microscopy? So always remember the triangle. IM. If we are looking for magnification, magnification equals to image divided by actual size. So this option is correct. And we would know that the second one is incorrect. So third option, actual size. So actual size equals to image divided by magnification. So number 3 is correct as well, and number 4 is incorrect. So your answer would be A. Question number 3. In which cell structures does DNA transcription occur? Right, so for transcription to happen, the organelle or the cell would need to have DNA in there. We know that nucleus have DNA definitely, and yes, chloroplast and mitochondria would also have DNA in them. The answer would be D. Question number 4. Which rows could correctly identify the nucleic acids present in 2 different virus particles in a sample of air? Now for the genetic material of the virus, it would always be either DNA or RNA. So it is impossible to have both DNA and RNA at the same time. Therefore, our option is only 2 and 3. Your answer is C. Question number 5. Test on 4 samples from a mixture of biological molecules gave the results shown in the table. Now, for the first test, we have Benedict's test. So Benedict's test tests for reducing sugar. If the reducing sugar is present, you would have brick red precipitate. So in this case, the color remains as blue. That means reducing sugar is not present. So any options with reducing sugar is incorrect. So let's eliminate them. So for the test of non-reducing sugar, before you conduct the Benedict's test, you would need to use acid to hydrolyze the non-reducing sugar to reducing first. Now in this case, after acid hydrolysis and neutralization, followed by Benedict's test, you do actually get a positive result, a brick red precipitate. That means you do have non-reducing sugar. This is also showed by the option. Burette test tests for proteins. So if protein is present, the solution turns into purple. So in this case, you get purple. That means protein is present. So you get protein. Your answer is C. Looking at iodine, so iodine tests for starch. If starch is present, you would get brown instead of yellow. So yellow is the original color of the solution. Therefore, no starch. Question number 6. One molecule of X is formed by a single condensation reaction, releasing one molecule of water. So X is formed by condensation Condensation is always building up two small monomers into a polymer. And you are only releasing one molecule of water. That means you are only forming one bond. So you are only connecting two monomers. So disaccharide would be correct because a disaccharide, di means two carbohydrate. A phospholipid would be incorrect because you have the phosphate group, the glycerol, and also two fatty acid chain. So if X is phospholipid, to form a molecule of X of phospholipid, you would require two condensation, one for each fatty acid. Same goes for a polysaccharide. Poly is more than two, therefore you would have more than two condensation reaction. You would be releasing more than one molecule of water. Triglyceride is the same as well. It is made of a mole of glycerol with three fatty acid chain. Therefore, you would have three molecules of water released. Now let's look at question number 7. The table shows some information about carbohydrate polymers. Which row describe glycogen? So glycogen is the carbohydrate stored in animal cells. Okay, therefore the function is storage. So let's eliminate C and D. Now the structure of glycogen do have branching, so they would be branch, and branching is all caused by the alpha 1,6 glycosidic bond. So this is correct. So our answer would be A. The diagrams shows 4 phospholipid molecules. Which molecule could contribute most to the fluidity of cell surface membrane? So the fluidity of the cell surface membrane, it depends on the fatty acid tails, specifically the unsaturated fatty acid tail that causes a kink. This is a kink. So what happens when there is kink is actually it can cause the fatty acid tail to be easier to separate. So the more unsaturated fatty acids that the phospholipid has, the easier to separate. So it would be more fluid. Your answer is D because the fatty acids in this specific phospholipid has the largest amount of unsaturated fatty acid. Number 9. Which properties of phospholipid explain why single layer of phospholipid added to water immediately form bilayers? The property of a phospholipid, that means they have the hydrophilic head and the hydrophobic tails. So when they come into contact with water, the tails are hydrophobic, so they would repel water molecules. So this is correct. The hydrophobic fatty acid chains repel water molecules, so the tails pack together, and also the non-polar fatty acid chain are attracted to each other by hydrophobic interactions. That is correct. So you have, if this is a second phospholipid, this is what it meant. The non-polar fatty acid chains, they will be attracted to each other through hydrophobic interactions. Yeah, and your phospholipid, the phosphate group from the phospholipid head is polar, so it will form hydrogen bonds between the water. So answer is A. Number 10. The diagrams show 3 examples of different bonds. Which bonds hold the tertiary structure of protein together? So tertiary structure of protein on top of the peptide bond and hydrogen bond that holds the primary and secondary structure together. You also have the interactions between R group. So you would have the hydrophobic interactions, the disulfide bond between sulfur, and also the polar bond between R group of opposite charges. So to hold the tertiary structure of the protein, we have, yes, this is definitely correct. Bond 2 is just your usual peptide bond. So this is found in primary, secondary structure, not just the tertiary structure. And here, bond 3 is actually, you have two sulfurs, so therefore it is the disulfide bond. So your answer is 1 and 3. Question 11. Which feature of collagen enables it to fulfill a structural role in skins and in tendons? Now, one thing every time you see collagen, always remember it is formed by triple helix. For each helix, the third amino acid is always glycine. So that forms the structure together. It's like a braid structure actually. Question number 12. The graph shows how the concentration of 4 components, 1, 2, 3, 4, of an enzyme catalase reaction change with time. So which component is the enzyme substrate complex? In the beginning of the process, you would have your enzymes. This part is the active site, and you have the substrate. When they collide, they form the enzyme substrate complex. So this is known as the enzyme substrate complex, where enzyme remains unchanged, and these are your products. So which component is the enzyme substrate complex? So I would say that the rate is similar. It doesn't change a lot, like it forming and reducing the enzyme substrate complex as well. So the answer here would be D. So amount stays constant over time. Question number 13. The table shows the Michaelis-Menten constant for 3 enzymes. So which interpretation of the information is correct? So in a graph like this, if the enzyme has a smaller value Km, the enzyme efficacy or affinity is higher because they can achieve the maximum velocity at a shorter amount of time. So always remember, the smaller the Km value, the higher the enzyme affinity. Okay? Right, let's explore each of the options. First one, enzyme C has a Vmax which is half that of enzyme P. 1.5, yes, it is half of 3.0, but notice what is behind. This is only 1.5 times 10 to the power of -2. This is -4. Therefore, it is not half of that. B. Enzyme C will reach Vmax in the shortest time interval. No, enzyme C would actually reach Vmax in the longest time possible, because this is actually 0.015. For P, it is actually 0.0003, and for enzyme F, 0.000005 millimole per decimeter cube. So enzyme C has the largest amount on this scale, So it would actually be this enzyme in comparison. Okay, option C. Enzyme F has the greatest affinity for its substrate. Yes, that is actually correct, because if you recall, the smaller the Km value, the higher the enzyme affinity. So in this case, enzyme F would have the smallest Km value. So yes, the enzyme affinity is the highest. D, enzyme P has a Vmax of 6.0 times 10 to the power of negative 3. That one we would not be able to know, because to find out Vmax, you would actually need to use the graph to plot the intersect between the x and the y axis. This is Vmax divided by 2, and then you times 2 to get Vmax. Okay, question 14. What explains how a signal molecule produced by one cell can be detected by a target cell? So your answer here for number 14 would be B, because the signal molecule always will have a complementary shape to the cell surface receptor. So every cell would have a cell surface receptor. There may be different receptors for different signal. So the signal is received when the shape of the signal is complementary to the shape of the receptor. So this is a very popular question that comes out quite a lot for topic 4. How a signal molecule can be detected, or cell signaling in general. Okay, question 15. Some enzymes are produced in the cell of the pancreas. The enzymes are secreted when required. Which process is used to transport these enzymes out of the cells? So secreted, exocytosis. Number 16. Plant cells were submerged in a solution with a water potential cell. Okay, in a solution. So the solution has a water potential less negative than found inside the cell. So the question says that the solution has a water potential less negative. That means the solution have a higher water potential compared to the plant cells. So what happens is that osmosis will take place from high to low water potential. So direction of water movement would be from high to low. So water will enter into the plant cell. And when water enter into the plant cell, the vacuole expands to become turgid, but it will not burst because the plant cell has a cell wall that can hold its shape and maintain the turgidity. Question 17. What will be present in each chromosome at the end of the G2 phase of the cell cycle? So in interphase we have G1 as G2. So G1, each chromosome only one copy. As DNA replication occur, so for each chromosome you would have a sister chromatid. So by the end of G2 phase, yes, they still are a chromosome with 2 sister chromatids. That means they have 2 molecules of DNA. Centrioles is not correct because your question is asking about chromosomes specifically. Centromeres will never duplicate, so that is wrong as well. So telomeres, instead of 2, you would actually have 4. So telomeres, you would have 1, 2, 3, 4. Those are the cap of the chromosome. Question 18. Which events listed are part of the cell cycle? Yes, interphase, metaphase, and cytokinesis. All of it is correct. So your cell cycle would include interphase, and then to your mitosis, which can be further branched out to your prophase, metaphase, anaphase, and telophase. After telophase is completed, the cell cycle enters to cytokinesis to divide the cytoplasm. Question 19. Which features of an organism are affected by a drug that kills cells that are dividing mitotically? So this is incorrect because cells do not get repaired by mitosis, so option 1 is wrong. Yeah, so your answer is actually D. So let's just fact check it. Cell replacement, yes, we do replace cells with new cells created by mitosis. Stem cells is also created by mitosis as well. For tissue repair, you replace the damaged cells with new cells that are created through mitosis. And tumor formation is also caused by cells dividing through mitosis as well. But this is uncontrolled mitosis. Question 20. The graph shows how the distance between sister chromatids curve P and the distance between chromatids and centrioles changes during part of mitosis. Which statement could be correct? So T, let's look at the first one. T to V represents interphase. I would say this is incorrect. So this is distance between chromatids and centrioles. So chromatids and centrioles would not be the largest, maybe somewhere towards the end. The distance between chromatids and centrioles should be very short in interphase. So if it's a cell, your centrioles only starts to travel during prophase. And during metaphase, where your chromosomes align in the middle. So this is the distance between centrioles and the chromatids. This is incorrect. So option 1, option A and C is incorrect. So we know number 2 is correct. T to W represents metaphase. Yes, so metaphase is actually the furthest distance where chromatids would be away from the centrioles. And then this is definitely anaphase because during anaphase, the chromatids separates and is moving towards the centrioles. So distance between chromatids and centrioles would definitely be shorter. In this case, the distance between the sister chromatids for curve P is increasing here. So the distance, I'll just draw in anaphase, your centrioles, and now in anaphase, A4 separate apart. So this is how your sister chromatids be separated. So curve Q is distance between chromatids and centrioles. It would be shorter than in metaphase. The distance between the chromatids, that is for curve P, is increasing. That is correct. So answer is B. Question 21. Which statement helps to explain the 2 sugar phosphate chain in a DNA molecule are constant distance apart? 2 sugar phosphate chain. So that is because each purine base is linked to a pyrimidine base. We have guanine to cytosine, adenine to thymine. So it is always one purine and one pyrimidine. Yeah, so you have purines and the pyrimidine. During DNA replication, what must happen before a newly added nucleotide is bonded to the next nucleotide in the strand. So during DNA replication, you have the exposed template strand, and the new strand comes in. So before the added nucleotide is bonded to the next nucleotide. So what it means is that because of complementary base pairing, as the DNA polymerase read along the template strand, you would have also new complementary bases. So the question is asking, before a newly added nucleotide is bonded to the next nucleotide, before this bond is formed, the phosphodiester bond here in green is formed. So what needs to happen would be complementary base pairing and hydrogen bond. So hydrogen bond between the bases here. So answer is 1 and 2. Option 3 is incorrect because phosphodiester bond forms after the molecule is bonded. Number 23. An antibiotic enters bacterial cells through a membrane channel protein P. Some bacterial cells have shown resistance to this antibiotic by acquiring a mutation which alters P, the channel protein. This mutation prevents the entry of antibiotic into the cell, so which conclusion can be drawn about how resistance to this antibiotic developed? So this is changes in proteins. We all know proteins are translated by mRNA. mRNA is transcripted by DNA. So what possibility can happen is that there is DNA mutation causing the protein to change its structure, the configuration. So let's look at option 1. The mutation changed the order of the amino acid in the gene coding for P. No, it would change. The mutation does not change the order of the amino acid. So this is actually, the mutation would change the nucleotide base in the gene. So gene is made of DNA. Therefore, the nucleotide base would be changed. Option 1 is incorrect. So let's eliminate. So we've narrowed it down to 2. Based on these two options, we know that option 2 is definitely correct. The mutation resulted in a production of P with tertiary structure. Yes, that is correct. The functional structure. Number 3, the antibiotic is a hydrophobic molecule and cannot cross the phospholipid bilayer to enter the cell. Well, that's why it needs the channel protein in the first place. Regardless of whether the bacteria develops resistance or not, it doesn't matter. Now, if the antibiotic is a hydrophobic molecule, it will be able to cross the phospholipid bilayer. Because your phospholipid bilayer, the middle part is the hydrophobic core, so only hydrophobic substances, small, non-polar, or hydrophobic molecules can pass through the four. Okay, let's look at question 24. What is the correct tRNA anticodon coding for the amino acid proline? So for amino acid, we have information about the amino acid and also the DNA triplet code on the transcribed strand. So we all know DNA will be transcribed to become mRNA, and the proteins is brought along by tRNA with the complementary anticodon. So if our code for the DNA, we are looking for the amino acid proline, the DNA base would be guanine, guanine, and thymine. Therefore, the mRNA would be complementary of that, which is cytosine, and adenine. So proteins are brought by the tRNA. Anticodon is complementary to the mRNA codon. Therefore, it is guanine. Instead of thymine, we have uracil. So your answer is D. Okay, question number 25. The table shows the observations made by a student about three different cell types, P, Q, and R, seen in a transverse section of a plant stem using a light microscope. So we have PQR, the ratio of the cell wall width to the whole cell width, and the structures observed in the cell content. Now, which type of cells are P, Q, and R? Let's look at the different options that we have. So we have companion cell, which is a living cell beside the phloem tissue. And also we have our xylem. So xylem are dead gibber. So always remember that. So that since they are dead, the ratio of cell wall width to the whole cell width would be much lesser. So P would definitely be xylem. Another way that you could know is that xylem are dead cells. Therefore, there will be no cell content. So option A and B could eliminate that. And for the phloem tissue instead, so we have our phloem tube with the sieve plate in between, and each phloem cell would be paired with a companion cell. So companion cell are living cells with all the organelles. So a regular pattern of circles in some cells, that would actually be the phloem sieve tube, because this is actually indicating the sieve plate, which is companion cell. Question 26. Water molecules are attracted to each other. This property is important in the upward movement of water in xylem. Which term is used to describe the attraction of water molecules to each other? That is definitely cohesion. Cohesion is the attraction between water and another water molecule. Adhesion is always between water and surface, like towards the wall of the xylem vessel. Hydrophilic is when a non-water substances is attracted to water and has the same meaning as polar. Okay, question 27. The diagram shows a model to demonstrate mass flow. So we have the partially permeable membrane here, concentrated sucrose solution within that membrane. We have water here around, and we have the dilute sucrose solution. So we have dilute, concentrated. Okay, and we are going to see how water moves. So in a plant, what are the structures W, X, Y, and Z? And what is the direction of flow of solution along W? Okay, now concentrated sucrose solution, this would be where the sucrose would be made. And in plant, photosynthesis happen at leaf. So leaf would be Y. Let's eliminate option, which is not Y. So leaves is Y. Now, so from there, the sucrose would be moving from the leaf towards the rest of the plant for storage. So Z would definitely be the roots, which is for storage. So both of the options have Z as root as well. Sucrose travels through the phloem. So phloem would definitely be where the sucrose travel. So W would be phloem. And movement of water is through xylem. Therefore, xylem is X. Direction of flow along W would be from Y to Z, because we are looking at the movement of sucrose. If it's movement of water, then definitely from dilute to concentrated sucrose solution. Question 28. The parts of the heart that control heart action are listed. We have the SAN, AVN, and Purkinje tissue. Which row is correct for atrial contraction and ventricular contraction? So, atrial contraction is always caused by SAN, and AVN is responsible to transfer that signal towards the Purkinje tissue, causing ventricular contraction. So let's look at atrial contraction. 29. The diagram shows part of the circulatory system in a mammal. So we have the direction of the blood flow. This is from the heart, oxygenated blood. From the heart travels through the rest of the body, and then we have oxygenated become deoxygenated blood, then they get collected back to the vena cava. So these are deoxygenated blood. Where is the blood pressure and the speed of flow the lowest? So speed of flow would definitely be lowest in the capillary. So capillary, number 1 here is the capillary. And the lowest blood pressure would be number 4, vena cava, main vein. The photomicrograph shows human body cells. What type of cells is X? So the small structure here is definitely our red blood cell. So here we have our white blood cell, and if you recall, white blood cell can be separated into lymphocytes and phagocytes. Now, in terms of the structure, always remember L for large nucleus. So the nucleus would be large. So for this one, this is actually a lymphocyte. Therefore, for phagocyte, they actually have lobed nucleus, like what you see in X here. So X is a phagocyte. Okay, question 31. The diagram shows some of the reaction of carbon dioxide when it enters the blood from cells in a metabolically active tissue, which reaction is catalyzed by the enzyme carbonic anhydrase. So, carbonic anhydrase catalyze this process. Number 32. The graph shows the oxygen dissociation curves for hemoglobin in animals that live in high altitude and animals that live in low altitude. So what explains the oxygen dissociation curve at high altitude? So at high altitude, what happens is that the hemoglobin will adapt to have higher affinity. That means it binds more easily to oxygen. So it helps people living in that altitude to adapt and to make sure that they can continue to live without any problem. Number 33. The diagram shows 3 features found in the mammalian gas exchange system. We have the ciliated epithelium, the cartilage, and also the goblet cells. And X is the similarities among those 3 cell types. Right, now, which structure of the gas exchange could be represented by X in the diagram? So if we look at the gross structure of the lungs, here you would have your bronchioles and your alveoli towards the end. So for cartilage, you would only find cartilage in the trachea and the bronchus, not bronchioles. So any options with bronchioles, we eliminated. Okay, and for ciliated epithelium and goblet cell, those are lining of the bronchus, bronchioles, and trachea. So your answer would actually be the trachea and bronchus. What are the short-term effects of nicotine on the cardiovascular system? That would be constriction of small arteries correct. So when you have small arteries constricted, that will increase your heart rate and blood pressure. So all of it is correct. Some features of the disease are listed. It can be transmitted by animals to another animals, including humans. One mode of transmission is by transfusion with contaminated blood. The causative organism can show multiple drug resistance, and the majority of humans who die from the disease are children. So, which disease has all of these features? So, cholera is not correct because it cannot be transmitted by animals to other animals. So, transmission for cholera is actually through contaminated feces, contaminated food, drinks, HIV and AIDS. Yes, it can be transfused with contaminated blood, but the organism did not show multiple drug resistance. Or people who die, they are not majorly children. So that is incorrect as well. Let's look at malaria. Yes, malaria is correct because first, it can be transmitted by animals to another animals. Yes, malaria can be transmitted by mosquitoes. One mode of transmission is by transfusion with contaminated blood. That is correct as well. The causative organism, yes, the mosquito can show multiple drug resistance. The malaria pathogen itself can also show multiple drug resistance. And yes, humans get the worst effect of that. TB is not correct because it is transmitted by droplets of coughing by the infected person. Question number 36. The following advice was given to a person traveling to a country where there had been an outbreak of an infection disease. So the advice was to cook food well and eat it hot, peel fruit and vegetables, drink only cool boiled water, and wash hands often with soap and cool boiled water. So which infectious disease would this advice help to protect against? When it comes to food, most of it is cholera because cholera gets transmitted by contaminated food and drink, water that has not been treated. Question 37. Which description of multiple drug resistance in bacteria is correct? Bacteria have DNA with resistant genes for several different types. Yes, this is actually correct. Let's look at the other options. Different bacteria species have DNA resistance for only one type. No, that is incorrect. They can have resistance for more than one type. C. Large number of one species of bacteria are immune to several different types of antibiotic. No, there would be, you can have multiple species of bacteria being immune to multiple types of antibiotic. D is incorrect as well. Many different species of bacteria are immune to not one, but many type of antibiotic. Question 38. Which description of a T lymphocyte is correct? They are only found in blood and secrete cytokines. That is wrong. They can be found in blood, but they can also be found along things like your lymphatic system and so on. They can leave the blood and accumulate at sites of inflammation. Yes, that is correct. They can leave the blood and secrete cytotoxins when exposed to bacteria. No, they don't really secrete cytotoxins. They circulate in the blood and always present antigens in response to infection. No, they don't always circulate in the blood. So T lymphocytes are only produced upon exposure of antigen. Question number 39. Where are antibodies found during an immune response? Yes, on the surface of memory cells, that is correct, and also in blood plasma. So you have antibodies present in both locations. Because this is during an immune response. If it's after an immune response, then only this one. So after or before an immune response. Last question. Monoclonal antibodies are used in the diagnosis of and treatment of disease. They are produced using a technique known as the cell fusion. So this is the hybridoma cells. Hybridoma is the combination of a cancer cell and a plasma cell. Always remember this. Okay, so that's it for today. Hope it helps. Comment down below if you have any more questions or what you would like to see more in the future. See you!